Hi students, antibodies are meant for specifically binding to epitopes of antigens such as pathogens which invade the human body. There may be many types of epitopes present on any antigen, for example, hemagglutinin and neuraminidase on the viral surface. Obviously, the antibodies binding to both of these must be mutually distinct from each other depending on the structure of epitopes these bind to. Accordingly, they originate from very different and unique B cell clones also. Therefore, these are termed polyclonal antibodies. On the other hand, antibodies are said to be monoclonal if and only if they possess a single antigen specificity, that is, bind to the same type of epitope of the antigen. Also, if they are produced from the clones of the same B cell. This justifies their name, monoclonal antibodies. These can't be produced in vivo within the live host body obviously because there are numerous epitopes of uncountable antigens present within the same. Koehler and Milstein devised an amazing method called Hybridoma technology, wherein loads and loads of monoclonal antibodies are successfully and indefinitely produced in vitro. This technology involves injecting the live host, let's say a mouse, with an antigen. The host responds to this in due course by producing antibodies. Now, the clone of B cells producing these antibodies is isolated from any secondary lymphoid organ, for example, the spleen. Thereafter, these antigen-primed B cells are put together with a type of cancerous plasma cells called the myeloma cells in the presence of biological fusion such as syndevirus or more popularly a chemical fusant polyethylene glycol. All this is done aseptically onto a selective culture medium called the HAT medium containing hypoxanthine, aminopterin and thymidine. This HAT selection depends on the fact that mammalian cells can synthesize nucleotides via two distinct pathways, the main or the de novo pathway and the salvage pathway that bypasses the main pathway. Aminopterin blocks the de novo pathway of the nucleotide biosynthesis, whereas hypoxanthine and thymidine serve as the precursors of all purines and pyrimidines respectively via the salvage pathway, catalyzed by the enzymes HGPRT, that is hypoxanthine guanine phosphoribosal transferase, and thymidine kinase or TK respectively. The B cells taken here are essentially HGPRT plus and TK plus, but due caution is exercised that myeloma cells chosen for hybridoma technology are essentially HGPRT minus and TK minus, that is, they lack the capability to manufacture their nucleotides from the precursors provided in the selection medium. Now, in this attempt of somatic cell fusion, three types of products can form. First, B cells fuse with B cells. These eventually automatically die out after a few divisions as these are the normal mortal animal cells. Second, myeloma cells fuse with myeloma cells. These can divide indefinitely but still have to die as they are unable to synthesize nucleotides due to the absence of the enzymes HGPRT and TK. Third and the last option is the combination of a B cell and a myeloma cell termed a hybridoma. This is the only combination that grows on HAT medium, that is, it gets selected since the feature of HGPRT plus and TK plus of the B cells enables them to synthesize their nucleotides from the given hypoxanthine and thymidine using the salvage pathway, and the immortal feature of the cancerous myeloma cell helps this hybridoma to live on eternally. For this brilliant and innovative work, Koehler and Milstein received a Nobel Prize in Physiology or Medicine in 1984. Indeed, monoclonal antibodies are their gift to humankind. They have not only revolutionized immunology and immunotherapy, but also the entire biomedical research. Guys, we hope this information adds value to your knowledge. Our endeavor is to explain tough concepts in an easy and interesting manner. I heartily acknowledge my worthy ex-student and now my YouTube teammate, Mr. Brahmjeet Dahiya, for the lovely illustrations of these videos. Together, we shall be bringing more such creations for your benefit. Keep watching and learning. Bye till next time.